dispersal, we think about islands as models, moving from island 1 to island 2 to 3 to 4. The Hawaiian Islands are this great example of what happens when you get islands of different ages. Really loud. Where islands form, over millions of years, and you get a new one every 4 to 5 million years. So the question is, when you go to an island, there's not one species of fruit fly, there's not one species of cricket or silver sword plant. There are lots of them. So where did they come from? Did they all disperse at different times? Did they get there as an ancestor and radiate on that island? And how can you tell? So the first model, there we go. So here's our old island, here's our new island, no new species. We think that islands generally start speciation with a new colonization event, and what happens is that species that lands there, that population that lands there, eventually develops enough differences that it becomes reproductively isolated. And when a North American scientist goes and looks at these flies and says, oh, you're different, now we would recognize the species on island 2 as a different species than the ancestor on species 1. Right? Then later on in the process, 1,000 years, a million years later, maybe you get a second event and a third event. So that what it looks like is that you've got a second colonization. Now, this ancestor that came from the same stock maybe as the original species looks different. And it can't talk to the new species, and it can't interbreed. So now you've got two species on this new island, but they came from the same ancestor, or the same ancestral stock. Okay? So this is dispersal generating new species on multiple species on an island. The alternative is that first event, the red event here, gets there, and then within that descendant island, speciation events happen. Maybe that population diverges and some of the species live on one fruit rotting plant, and another live on a different species. Maybe one of them ends on the east side of the island, the other's on the west side of the island, so there's a physical barrier. Could be all kinds of mechanisms, but you get a cloud of species that come out of that ancestor, that first invasion event on island 2. And we see lots of evidence for this. This is a lineage of crickets that lives on the Hawaiian Islands, La Paula. And you see a phylogenetic pattern that looks kind of like this. And what you should pull away from this is that each of these islands, remember that here's our old island, Kauai. Oahu's old, but not quite as old. Maui and Molokai are intermediate. Hawaii is the new island. You should see a pattern that looks something like this. All of the species that have an H are now found on Hawaii. What's their common ancestor? Well, it goes back to one Hawaiian cricket. That means speciation happened on the Hawaiian island, the island of Hawaii. Right? And that ancestor shared an ancestor with a cricket from Maui, right? One of the older islands. Right? All the crickets on Maui trace back to a Maui ancestor that shared an ancestor with a cricket from Kauai. Right? So if you go back in time on the phylogeny, you're going to get ancestors from the older islands, and all or most of the species on the current island, so here's Maui, M, all come out of one ancestor from Maui. There are going to be exceptions, right? If you look in Hawaii, here's one that ends up on Maui, even though it's from the Hawaiian clade. So there's some back dispersal. Right? You're never going to find a perfect pattern. But that phylogenetic pattern fits speciation on an island after a single or few invasion events, as opposed to dispersal over and over and over again. All right? So let's start with a question. It should have been on last time. And we'll see if I've actually got this thing functioning right today. So this is a question about reading a phylogeny and inferring patterns of speciation. Can you tell, A, C, Can you tell from the phylogeny what the pattern is of speciation? If we had a phylogeny, which of these phylogenies supports the idea that these invasions from 1 into 2 are the source of all these origins? So the question is, does one of those patterns or the other, or neither, support this idea that the 5 or 6 species we see on island 2 came from repeated dispersal events? Dead silence. You don't want to talk to each other. It's like, no, it's early. It's 10 o'clock on a Monday. What I see from the numbers arising is that most of you are either still asleep, don't know where your clicker is, or can't be bothered. So apparently it's not quite as obvious as you think. All right, most of you have tried this. So what do you see as the difference between these two trees? So here's the clue. Despite those of you who always like this answer, could produce either pattern, that's never the right answer, right? That's like us trying to come up with E as a choice, right? We wouldn't ask if there wasn't, almost never do we ask if there's not an obvious pattern. So what's the difference between these patterns in branching? What jumps out right away? What is true about the one on the right? That's a question. Somebody help me out. What do you see that's different over here from over here? This is the point where I get awkward and I just do the, until somebody answers. Come on, like what is the topology as a pattern? What's the topology over here? Where are all the 2s? Are they separated? No. Are they together? What's the common ancestor of all the 2s? Is it a 1 or a 2? Two, thank you. Let me get my dental tools out. All right, how about here? Does 2 have a common ancestor that's a 2? No, 1. All right. So the island 2 has descendants that come from where? 1. Okay? So, which pattern, what does the pattern look like if repeated invasions from island 1 were the source? Probably over here. All of the species, or many of the species, most of the species on island 2, came from 1. Right? So this is important because it's a way you can look at a reconstructed phylogeny, the relationships between species, and figure out where did they come from. Is this crickets from Oahu keep moving over to Kauai, or did one cricket lineage from Oahu land on Kauai and then radiate out? Okay. All right. Think about phylogenies and speciation patterns. So here are the words that we just went through, just so they're on the notes. Right? So repeated invasions are going to look like the species on island 2 have ancestors from island 1. Right? So we're going to find multiple branches. Whereas if we have speciation in situ on that island, we're going to find one most recent common ancestor that all of those lineages come from. So that's the cricket picture I showed you a minute ago. I'm not showing you another alternative, which is that there's vicariance within that island, right? Then you would see these branching patterns 2 by 2. Right? Okay, so let's move along. So we had vicariance, we had dispersal. There's another way that geographic factors can influence speciation, and this is usually called isolation by distance. The difference here is that now we no longer have a barrier, a border, right? So we don't have a wall in the isle that separates this side of the class from this side. We just have distance. So over here on the far aisle, it's really hard for those gametes to get all the way over here. And you might think about it like, you can have these contiguous distributions, meaning unbroken. Right? You could have mating events that go, four rows over, 4 seats over, right? And the next generation, maybe another couple seats, and eventually an allele that was over here after many generations could move, like a little hopscotch. And then another couple generations, and eventually after like 30 mating events, an allele that was over there could end up over here. Well, take that to a continent, right? So this is a map of subspecies of garter snakes. It could be anything. All you really need to pull out is, here's North America. You've got different groups on the East Coast than on the West Coast. These are named subspecies. They look a little bit different, but not so different that people think they're different species. So how does a snake in Virginia get alleles all the way to California? Why do we think they're the same species? Well, maybe they eventually get to West Virginia and they mate, and then 20 years later, some of those descendants mate with snakes in Ohio, and eventually they mate with snakes over in California. All right? So that means that there's a continuous distribution of allelic exchange. Gene flow's moving across that whole group. But if I took a snake from California and a snake from Virginia, they may not mate because they're so separated in space. So how do you deal with that as a species? Along each step of the way, they're close. But at the ends of the distribution, they seem to be really different. Why is that important in a sort of real-world biological case? You get these weird geographic patterns that generate a thing called ring species. And this is one of those things that biologists love to argue about because they have particular definitions, and does this really fit a ring species? But the basic idea works pretty well. And it's like the physical manifestation of that general model that I showed you last time. Right? We start with an ancestral group. It separates out into 2 lineages. They start acquiring genetic differences, and they come back together, and they're isolated. And they're isolated in this case because along the way there's some barrier in between, a mountain range or a desert or a body of water, and they cannot exchange genes in each of those groups. And as they move biogeographically over time, let's say north to south, they eventually get to the end of that barrier, they come around, they touch, they come back together, and now they can no longer interbreed because they acquired enough genetic differences. And yet all along the way, you could say, oh yeah, they interbreed, and you could make that whole loop, except where they close the circle, where they close the ring, they can't talk to each other anymore. So a great example of this, and there are a few, but this is probably one of the best worked out, is a salamander called Encetina. And you can see they're kind of a sexy, well, for those of us who like salamanders, these are sexy salamanders. They probably came from a northern refuge. As the glaciers receded, they moved onto the coast, they moved down. California has this weird geology where there's a central valley that's basically a desert. And then there's a mountain range, the Sierra, that comes down here, and the Cascades and coast ranges down here. And those are similar in habitat. They're cool and kind of humid, and salamanders like that, right? But they don't like desert. So what you had was this invasion of salamanders down from north to south. One group branched off and went down the Sierra. One group branched off and went down the coast. And now they're back in contact down here. And if you ask, will they mate with each other? Basically, they're happy to mate all along the way. Except when you get down here into Southern California, where you get this black and yellow one, Cruciator, in contact with the Schultzi. And you put those two together and they look at each other like, yeah, what am I going to do with you? There's no recognition of potential mating between those two things. And yet all along the way, there's not very much genetic subdivision. There's a little zone of hybridization here where this yellow-eyed Xanthopithecus species gets across the desert into the foothills of the mountains. And so you've got a little tiny bit of gene flow. But the basic idea here is that if you map the genetic distance, you'd find that each of these is pretty close, pretty close, pretty close, and you get down here and they'd be very distant. So not too long ago, a group at Berkeley worked out this phylogeny. Here's Oregonensis, the red one, that's the ancestral type. Here are the southern coastal ones, yellow and green. They kind of branch off. You get a different branch out here for the Sierra, and where they come back together, purple and green. Here's green, purple's all the way out here, so pretty distant, right? There's a good example in the book of warblers in, I think in Mongolia, that has the same sort of dynamic. We see this not uncommonly, but the geography has to be just right to get the idea. But it fits that whole model of isolated groups acquiring differences. When they come back together, they can't meet. But we see it in space rather than time. I think that's why it's so appealing. On a smaller scale, and this example is partly because it's local, you can see this in just linear, very short linear distances. So this is a salamander called Desmonathus that lives on mountaintops in the Appalachians. This is work that was done by Steve Tilley and Steve Arnold, maybe 20 years ago now. So these guys live on the tops of mountains like Mount Rogers and in the Smokies. And if you look at their phylogeny, you look at their genetic distance, this is a map. Here's the Virginia-North Carolina border. Here's Tennessee. This is the Appalachian Smoky Mountain line. You've got groups where they come close together. They're pretty close genetically. As they are further apart, so the southern one up to Mount Rogers, you get huge genetic distances. Right? So if you plot that out, what do you see? Bottom graph, plot geographic distance to genetic distance, nice positive correlation. Not a huge surprise. Salamanders don't move very far. They don't like to go to the lowland. So in our current climate, they're stuck on mountaintops. They become more genetically isolated. Right? Second one, here's genetic distance now. So how genetically differentiated are each of these mountaintops? Tilley and Arnold and Varel, actually Paul did all the work for this. So he put salamanders in boxes together and asked, will you mate? And they ask, how close to mating will you get? And salamanders do this weird, like 8-hour courtship where the male walks along and he wags his tail and the female gets on and she follows the wagging tail and he turns around and he slaps his head on the female. And this group has these, this is more than you needed to know. This will not be a mate. It has premaxillary teeth that have during mating season, grow into basically fangs. And the male whips around and slaps the female and cuts her, like scrapes the skin, and has a gland with, they're called mating compounds, mating pheromones, that increase her receptivity. So the male whips around, slaps the female with these fangs, rubs this gland on her back, and this goes on for hours. And so they've developed this isolation index that says how far in that sequence do they go. Do they do the tail straddle walk, or does he slap her, or does she respond to the slaps, and so on. And so the higher the isolation coefficient, the less far in that process she goes. Right? So the further their genetic distance, the more isolated they are, and that maps up with geographic distance. So if you remember nothing about tail straddle walks, what you need to know is that they are more isolated the further apart they are genetically. So the more physically distant they are, the more genetically differentiated they are. And the more genetically differentiated they are, the more behaviorally isolated they are. Why? Well, it could be that they are behaviorally isolated and therefore they can't mate, and so they became genetically distinct. Or it could work the other way. They became genetically distinct, and now there's selection against interbreeding. We'll come back to this in a minute. And so there's been selection favoring the behavioral isolation. So the correlation doesn't tell you which direction the causal arrow goes, but it's there. And so we'll look at why we might expect them to evolve isolation in a minute. Okay. So everything there is focused on spatial differences, geographic distance as an isolating mechanism or a physical barrier. And I said at the beginning of this section that this guy, Ernst Mayr, made a big deal over the years about that being the only way species could form. So unless we had physical barriers, you couldn't really come up with any way to isolate two groups long enough to accumulate genetic differences. We now know that that's not true. So we're going to focus a little bit on sympatric mechanisms, but really it's the same process. It's just that something goes on without that crystal separation that creates rapid isolation. What's different about allopatric and sympatric, that really has to be different, is sympatric speciation has to happen faster, right? Because you don't have a long time for differences to arise, for isolation to arise. So the mechanisms we'll think about have to do with rapid speciation, right? So isolation can come about through all kinds of things. We talked about those. I'm not going to belabor them. I draw your attention to things like that mechanical isolation example with snails. That is instant, right? It's one maternal effect gene. All of the babies from one snail acting as a mom have this changed chirality. Now they can no longer mate with the other offspring from that entire species. That's instant. This is, I hate the term, and I'm going to use it anyway. For a while, people were calling these magic traits, right? Because they create the difference and the isolation at the same time. Right? Chirality is kind of a magic trait because it's a single gene that instantly creates reproductive isolation. Okay. Another form of sympatric speciation, maybe in some groups, maybe the most important, and I'm going to give you an overview, and if you want to dig into it, ask Brittany, because this is what she studies, is polyploid speciation. And I'm probably going to make a hash of notation here. So you guys are basically familiar with this. You get a meiotic accident, you get non-disjunction, or hybridization retains chromosomes from both parental types, and you end up with a hybrid that has a different chromosome number. Well, right away, that hybrid can't mate with either of the parental types. Right? So, an example here is, it happens all the time in plants. This is a Gilia. So here's a parental type with 9 chromosomes. Here's a different species, very different plant, 9 chromosomes. Hybrids retain both sets of chromosomes. They now have X equals 18 chromosomes. Right? What happens when Gilia major tries to mate with Gilia minor? They have the wrong chromosome number. Right? They have an uneven number. So the male and female gametes can't form. Right? So why does this thing even persist? That's a good question, and we'll tackle that in a second. But for a long time, people focused on this as some weird thing plants do. Right? So why does it work in plants? Well, a plant gets this weird chromosome number, it makes lots of seeds, and they can self. So they've got a group of other individuals they can cross with. But we know that's not the whole answer, because sometimes there's self-incompatibility, and we also know that animals do this too. So I'm going to bring you back here. I'm going to bring you back to animals in a second. So why do the polyploids last at all? This is something that drives me nuts. So, okay, you've got these two plants, they cross, they get a 2x. So n is the somatic number. Everything's 2n that we're worrying about, diploid animals. And the x is the number of gametic chromosomes. So if you have 2n equals 4x, that's a tetraploid. So why do those 4x individuals persist? Is there something good about being a polyploid? It turns out, yeah, there is. And some of this, the first time I heard it was like, really? There's pretty good evidence now that polyploids are different enough that they can invade new niches. I'll just show you an example of this in a second. So there's something about that genetic combination that allows them to invade a habitat that's not occupied by either parental type. Right? So that's sort of a selective argument, that there's a new niche available. In plants at least, this is the part that I thought was weird. Polyploids have bigger cells, they're bigger plants. That means they can do things, they have more DNA, so they can do things like make more defensive compounds, right? They're proteins, so they make more good stuff. So polyploids might just have an advantage because they're bigger and make more. In an evolutionary sense, because they have more DNA, they have more gene copies, they may be able to evolve in a different way. So I think you guys talked about duplication and neofunctionalization as mechanisms of adaptation. Think about those two groups that have 9 chromosomes each. They come together, now they have 18 chromosomes. Well, a lot of those genes are duplicated. That means you got one copy that can do the important thing, the basic organismal function, work in the Krebs cycle. Right? And then there's another copy of that gene that could either be silenced, thrown out of the genome, or allowed to evolve a new function. So they may have adapted pathways that are not available to non-polyploid species. Right? And just to make this point that it's not plants only, back to this thing you guys are all tired of now, the gray tree frog. The tree frog, I played these songs last time, this kind of bird-voiced one, versicolor, that's a tetraploid. Chrysosceles, the one that's a little more like a robot, is a diploid. We know from work that was done by a woman in Texas about, 8 years ago now, that these tetraploids are not a single polyploid origin. So this is wild. I told you that polyploids are like this, you make a polyploid, they can only mate with each other, and that's instant isolation. She traced through this versicolor cloud and found that there are at least 3 hybrid origins of this Chrysosceles mating with some other group and creating a tetraploid lineage. 3 different times. 2 of those groups are now extinct. We can't find that part of the genome in any extant frog anywhere. But we can see that it's different from the Chrysosceles genome, and we can see that it's different from the other hybrid origin. And yet those tetraploids came together and could mate. And so now what we recognize as versicolor is this cloud of tetraploids that has an origin in 3 different hybridization events. So the hybridization didn't create perfect isolation, it just isolated it from the diploid parental stalks. Okay. Hybridization's bad most of the time. Right? So I want you to try and think about hybridization as having 3 possible outcomes. I've tried to emphasize for 2 days that if you form a hybrid, you don't have isolation and you can't get 2 new species. Okay? That's sort of the dogma. Right? So what happens? Here we've got a blue group and a red group. If they cross, they create hybrids. The hybrids are less fit. They're genetically incompatible. Or they have low ecological fitness. And so selection should favor mechanisms that prevent hybridization. This is called secondary reinforcement, and I'll come back to this in a second. But the thing that's important to remember here is that it's called secondary reinforcement. I'll reiterate this in a second. Because it happens second, it can't happen first, and it reinforces isolation that already exists. So essentially what we're imagining is that we got isolation, the 2 groups formed our incipient species. When they come back together, the offspring are not as fit. So what is selection going to favor? It's going to select parents that won't waste their genes creating an unfit offspring. So isolation is secondarily reinforcing isolation. So it's always going to be prezygotic, and it's going to reinforce postzygotic. I'll say this all again. All right. That's outcome 1. Outcome 2 is that it's actually a good thing. And it can be a good thing in the sense of creating a new lineage. It can be a good thing because it creates a polyploid event or new gene combinations. But for whatever reason, this cross between the two parental stocks leads to a third lineage. Both parental types persist, but that third group also persists and becomes isolated from either parent. And I'll show you an example of this. But we now, this is actually a pretty recent, maybe 10 years, appreciation that this is actually a fairly common source of new species. And the third version is what we used to think was the only possible outcome of viable hybrids, which is this ugly graphic here where the two parental types come together, they form hybrids, but they only form hybrids where they come together. So the isolation isn't perfect. The parental stocks are still pure, but there's what we usually call a stable hybrid zone, meaning that there's some part of space where those two groups come together and they mate, and they make viable offspring, but those viable offspring don't move very far out of the zone. And maybe they can back cross a little bit, but the parental types still persist. So we see this flock of organisms in usually a tiny band in space that form hybrids, but they don't go anywhere evolutionarily. They don't become isolated from their parents. But it may persist for a long, long period of time. So these are the kind of three extremes of hybrids. All right, so I tried to emphasize the secondary reinforcement thing. It's a really important idea in speciation because what we think happens is if you think back to the general model, we accumulate differences, they come back together, they're genetically incompatible. That means hybrids are bad in that situation. And any adult parent, potential parent, that wastes its gametes or its mating effort or its investment in eggs, so gametes, on a hybrid offspring is going to be less fit than one that makes the right choice and mates with a pure parent and produces offspring that are going to have higher fitness. Okay? So, if hybrids have low fitness, selection should favor mechanisms that prevent that formation. How does that happen? Remember, pre- and post-zygotic mechanisms? It has to be a pre-zygotic mechanism that's favored through secondary reinforcement. Okay? So only pre-zygotic isolation mechanisms could evolve through secondary reinforcement. You can still have a post-zygotic isolating mechanism. It's actually driving the pre-zygotic mechanism. So these are important directional concepts. But only the pre-zygotic mechanism can arise from that secondary reinforcement. What's the other thing that has to be true there? It's secondary reinforcement because they had to come back together. So if we think about the island examples, you move crickets from Oahu to Hawaii. The ones in Hawaii become genetically distinct, and if you brought them into the lab and let them mate, they produce less fit hybrids. Are they undergoing secondary reinforcement? Shake your heads with me. No. Because they're not in contact. So there's no selection for the crickets on Oahu and the crickets in Hawaii to become isolated. No secondary contact, no secondary reinforcement. All right. So do we know that this happens? All the time. This is an example that's worked out in the book in great genetic detail. Have a look at it. It's Mark Rauscher and Robin, what's Robin's last name? Hopkins, I think. Anyway, flowers, Texas. Phlox dromidae, Phlox cuspidata. Right? They look like phlox. If you know garden plants, they're like a mat with they look like a flower, like a nice blue violet or kind of pink violet flower. Both species look really similar where they're allopatric. So it's the yellow and pink ranges. They look like a bee attracting phlox. Okay? where they come together and their zones overlap, where they're in sympatry, Drummond's flocks changed. It moved away, right, in phenotypic space. And now it looks much more red. And what Rauscher and Robin, whose last name is escaping me, worked out was the genetic pathway by which that difference arose. It's a really cool story, where the Drummondii in this overlap zone became red instead of pink blue. But the reason is because the hybrids have low fitness, and so the Drummondii that have the highest success are the ones that don't attract the same pollinators that pollinate the other flower. Right? So the pollinators moving from Cuspidata to Drummondii, it's exchanging gametes with the wrong species. So you're wasting your effort on a hybrid. Right? So that's bad. All right. This is another example of secondary reinforcement that's really well worked out, and it's one of the reasons to bring it up is that it emphasizes that it's pre-zygotic, not pre-mating, right? It's not pre-copulation, it's pre-zygote formation. This is work that Daniel Matute did for his dissertation in Chicago. He's at UNC now. It's a story of Drosophila, fruit flies that live on an island off the coast of Africa. There are places where Yakuba, in red, lives all by itself at high elevations, and Saint Tomé at low elevations lives all by itself, and they come together at the intermediate elevations. And they form hybrids. And the hybrids have this pattern that we talked about a lecture or two ago called Haldane's rule, where the males are sterile. So they make perfectly good-looking flies, except that the males are all duds. Right? The females are perfectly fertile, but the hybrid males are sterile. Right? So what Daniel was trying to figure out is there any selection because of that male sterility in the hybrids for secondary reinforcement, and can that happen post-mating? Right? Because we know that Yakuba and Saint Tomé are perfectly happy to mate with each other. So what he did was he took females from the zone of allopatry and females from the zone of sympatry. What's the difference? In allopatry, those females evolved without ever interacting with the other species, and probably don't have any reason to have selection favoring mechanisms that are the result of interaction with Saint Tomé. And then he took this other group, the sympatric females, who do evolve in that zone where they might make the wrong mate choice, and ask, are they different? And what he finds is females from allopatric crosses, so this place where they're not evolving in sympatry with a similar species, they make about 75 to 80 eggs after mating with the wrong species. So you take one fly, put them together. Female from allopatry makes about 80 eggs. You take a female from the zone of sympatry, you do the same thing, give it the wrong species, makes about 30 or 40 eggs. Why? Because they're perfectly capable of interbreeding, of fertilizing the eggs. It seems like she's holding back eggs for another event. So what Daniel did next was to take females and give them two choices. So first he gives her the wrong species, and then he lets her mate, gender pronouns. The flies get a second mate, which is a conspecific. So they mate the first time with the wrong species, the second time with the right species. What do? In allopatry, it doesn't matter. A female comes from allopatry, she mates with whoever, she makes the same number of eggs from the wrong species male and the right species male. You go to the females that have evolved in sympatry with the wrong species, with the other species, she's holding back. She is somehow able to use the sperm from the first heterospecific mating much less often than the right male from the conspecific mating. Whether she's storing the sperm and sorting them out internally, or whether there's some behavioral cue, that's not clear. But she's producing less than 25% as many eggs using the heterospecific sperm. So this is really good evidence that selection in sympatry is favoring some isolating mechanism that's happening post-mating, but pre-fertilization. She's not wasting her eggs on sperm that are going to create a less fit offspring. And that's the story of secondary reinforcement. They come to into contact, selection favors those kinds of mechanisms. Okay. So, a question. If you follow that kind of story, which of these is likely to result from secondary reinforcement? There are like 200 of you out there. It is quieter than my house with one 16-year-old boy. Oh. Ooh. No, talk, really talk. Really talk.
Yeah, I could be.
Can I draw your attention to the question? What does the question say? All right, read that carefully and then see if your answers change. And talk to each other. Okay, let me try this again. Let's look at the question and see if your answers change. Secondary reinforcement is most likely to result in, that means what's going to come out of secondary reinforcement. It's not saying what does it result from, meaning the cause of secondary reinforcement. Does that change the way you're thinking of it? I'm asking what things evolve as a consequence of secondary reinforcement. And what I'm seeing is you guys are like randomly changing your questions. Like you're like, now it's just like A, B, B, C. Okay. All right. Let me try and clear this up. Raise your hand if this is a pre-zygotic isolating mechanism. Hybrid vigor? Hybrid sterility? Hybrid inviability? Pre-zygotic? Temporal isolation? Okay, so at least 2/3 of you won't raise your hands. What kind of mechanism evolves through secondary reinforcement? Pre? Post? Pre? Pre? What kind of mechanism evolves from secondary through secondary reinforcement? Pre? Pre? That should make you all like stand up. Like physical investment. Temporal isolation is the answer. Right? It could have been any kind of pre-zygotic mechanism. The important point here is that the post-zygotic mechanism creates the selection that results in a pre-zygotic mechanism. If you're going to memorize any one thing, and I don't recommend that, that would be a good phrase to memorize. Probably not, but good point. Post-zygotic isolation creates the selection against forming hybrids that results in pre-zygotic isolation. Got it? So you have to think about that direction, and that's secondary reinforcement. Okay, let's see. I can get through hybrid speciation as quick. So I told you that one of the three possible results is hybrid speciation, that cross generates something that becomes its own new lineage and is isolated from either parent. Okay? It's isolated from either parent because it's favored and it has the offspring between the hybrid and the parent, what we would call a backcross, have lower fitness. We now know this happens all the time. And one of the mechanisms we think is important in creating hybrid species is we get new genetic combinations. Right? Think about our homozygote cross. You cross them. What do you get? You get an F1 heterozygote. Well, imagine doing that in all the loci in a genome. We take two pure lines, we cross them, we get all these new combinations. There's lots of stuff there for selection to work with. The best worked out example, I think, is the stuff that Warren Riesberg did with sunflowers. Been ongoing for now maybe 20 years. He has a very cool sunflower called Anomalous, Helianthus anomalous, that lives in dunes, so really loose soil, sandy soil, not a lot of water, not a lot of nutrient. The hypothesis is this thing came from a cross of these other two species, Annuus and Pediolaris. And what Lauren and his lab have done over the years is do some genetic mapping and some experimental crosses, and it shows that the genome of Anomalous, is made up of chunks of Annuus chromosomes and Pediolaris chromosomes. So if you looked at the phylogeny of sunflowers these days, you would see this one species that's a branch that has no single ancestor. It has two ancestors. How do you know that? Well, the idea, Lauren's first idea, came from just the observation of space. Right? If you look at the distribution of where Anomalous is in black, and the two purported, the two maybe parents, it's where they overlap. And it's only in these green habitats. And what he did over the years were these replicated hybrid lines in the greenhouse. So these are not out in the wild. It's in the greenhouse, crossing and then back crossing and crossing the F1s. And he found that the anomalous chromosomes looked a lot like the experimental chromosomes. And they had traceable elements to each of the kernel types. So you could use these markers to identify which chunk of the chromosome came from Annuus and which chunk came from Pediolaris. And if you cross those guys in the greenhouse, they come out with pretty much the same chromosome map as the ones in the wild, even without the selection from the natural environment. So it means there's something about the way those blocks fit together and function as chromosomes that favors particular blocks in that hybrid order. And now, last slide, he's actually found that that same cross spun off 3 different species that are all in weird marginal habitats. So it's not just one, it's not even just one from a cross. They can create species from, multiple species from the